Good afternoon, everyone, and happy Thursday. Thank you all for joining us today for the HMSC Research Seminar. My name is Yi-Chung Chung. I'm the Academic Program Manager here at Oregon State University's Hatfield Marine Science Center in Newport, Oregon. I'll be your host for today's talk. This is a hybrid event with folks attending both on-site and online. Roseanne is our hybrid support today. Please let her know if you are having any technical issues so she can assist or let those of us on site know. For those online, we have your mics, camera, and screen share off. Let me just double check that I've done that real quick. Mm-hmm. 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 Okay. There we go. One last thing there. Hide this. And let's see here. We ask that you ask any questions online by letting us know either with your virtual hand or typing it in the chat. We will answer your questions at the end of the talk. We may also call on you to read out your question if you'd like. You're also for folks on site, please raise your hand and we will bring the mic to you so you can ask your question, for those folks online to be able to hear the question. We'll be recording this event and it will be posted on the HMSC past seminar page in a few days. We also have a YouTube channel that you can also look at our past seminar recordings. This is our last seminar for 2023. So please join us again when we start back up after the holidays in 2024. Let me introduce you to today's wonderful speaker. Dr. Cheryl Barnes is an assistant professor in marine fisheries at Oregon State University and the principal investigator for the Integrated Marine Fisheries Lab as part of the Coastal Oregon Marine Experiment Station, one of OSU's several experiment stations here at Hatfield Marine Science Center. She also serves as the Oregon Department of Fish and Wildlife representative on the Scientific and Statistical Committee for the Pacific Fisheries Marine Council, Fishery Management Council. Cheryl got her BS in biology at San Diego State and her master's degree in marine science at San Jose State University's Moss Landing Marine Laboratory. She received her PhD in fisheries from the University of Alaska, Fairbanks. Before coming to OSU in 2023, Cheryl was a research scientist at NOAA's regional office in Juneau, Alaska. Cheryl's work at OSU focuses on better understanding population and community dynamics of groundfish species in the North Pacific. Cheryl is particularly interested in developing scientific products that can be used to inform stock assessments and ecosystem-based fisheries management. Dr. Cheryl Barnes, the floor is yours. Take it away. And please welcome our speaker.
Okay, can you hear me? Yay. Okay, that was like my whole intro, so thanks, Yi-Chung. So the title of my talk is Navigating Uncertainty Through, oh, that is fun. Can we turn that down just a skosh? Thanks. Oh, warmer light is better, thank you. I don't know. This is good. Yeah, this is great. Okay, so the title of my talk is Navigating Uncertainty Through Stakeholder Engagement and Integrated Approaches to Fishery Science. So I'm going to talk more about collaboration, a little less about science. So in any of the examples that I provide today, if you're interested in learning more, let me know. I'd be happy to chat with you about it. Okay, great. So I want to start off by talking a little bit about what I see as the benefits of collaboration. So when we think about collaborative fisheries, I think a lot of times we go to the low-hanging fruit, where we want to collaborate because we can leverage resources, we can reduce costs, and we can increase sample sizes. That is all great. I have benefited from collaboration in all of those ways. But I would actually, because words matter, call that cooperation and not true collaboration. So I want to talk about what true collaboration means to me in fisheries science. And there are a ton of benefits to collaborating with folks across a variety of areas. So academics, agency scientists, resource managers, and fishery stakeholders are most of the folks I'll be talking about today, in addition to students. From the fishing side of things, one of the biggest benefits is that there's just a wealth of knowledge out there in the local recreational and commercial fishing communities. I have benefited a lot from the knowledge about life history, movement, and habitat use of groundfish from California to Alaska. Another benefit is that the folks who have been fishing, mostly, I guess I'm thinking about like charter fishers, guided sport fishing lodges, commercial fishermen, they have this long time series of information in their brains. And so there's a lot that we can learn from that, especially when those of us haven't been in fisheries for 3 decades or more. And this is probably an example that you all are familiar with from fillet stations all over the country, where we used to see these really big fish, and this was more of a commonplace catch. This is, I think, a photo from the early 90s. This is a collaborator of my father. But now what's a common catch is the image on the lower right. So we still see occasionally Pacific halibut that exceed 100 pounds in Southeast Alaska, but that is much more rare than it used to be. Okay, and then there's a couple other points that I want to get into before we talk about some specific examples. One of the benefits that I have seen with collaborating, again, with stakeholders and resource managers, is that we can together develop questions that are of interest to everyone. There is a place for science's sake. That is just not what I do or what I am interested in. I really want to see what questions folks have out on the water, and that those sorts of conversations to develop research questions have been really useful for me. Also, there's a lot of opportunity to gain insight and expertise from these folks. I mentioned that just a second ago, but I wanted to highlight some of the areas that maybe we don't often think about, which is using that insight and expertise to really develop and fine-tune hypotheses after you've identified the questions of interest, design effective sampling protocols. So again, if your sample size is king or queen or monarch or whatever you want to say, you need to be able to find the fish, and these are the people that know where they are. And then one of the key pieces that I've brought into my work is bringing information back to these communities to try to understand what we've seen and get their perspective, and that's really been very valuable for all of my projects. The other thing is that we can create a shared understanding of the resource. This is particularly important in fisheries, where we share this resource, and there's often animosity, more in some regions than others, between scientists and fishing communities. And so when you work together to develop a shared understanding of the resource, I think you can figure out how to move forward better. And then engaging those that are most impacted by management decisions, I think, is an ethical responsibility that we have in fisheries. Okay, and then there's a couple points where I think that we can maximize some of these benefits of collaboration. And that is really facilitating these ongoing conversations at all stages of a project. So again, from identifying research questions through to synthesizing and interpreting results. And Richard Yamada is like the gold star of the fisheries stakeholder collaborator. He has such an inquisitive mind. He's always looking at what's the next project we can work on. Right now, he's trying to put together a squid fishery in Southeast Alaska. So he's just, if you were to go on this guy's Facebook page, it's squid everywhere. Before that, he was really interested in competition between halibut and arrowtooth. That I'll talk a little bit more about, but he was really engaged in all of those aspects that I talked about earlier. Identifying the question, figuring out how we can actually go and sample fish, trying to understand what it all means when you're dealing with a really complex ecological question. And then he's been involved as a co-author on the paper that came from this project. So another thing I'd like to state here is that whenever it's possible, I think it's important to think about these folks from the very beginning when you're writing grant proposals, to be able to compensate them for their time or pay for supplies that they may need, things like that. And then offering an opportunity to co-author publications if it's appropriate to do so. I think providing that opportunity really is a way to engage people throughout the process. And again, Richard Yamada is the gold standard for that. Okay, so now I'm going to go through a few examples of collaborative research that I have led. There's kind of 3 key points that I will hit on. So I'll talk about a project that was really used to inform a stock assessment in California. I'll talk about another that was intended to promote EBFM, or ecosystem-based fisheries management, in Alaska. And then another that is intended to support climate-ready fisheries, again in Alaska. And I put climate-ready fisheries in quotes because I still don't really know what that means. We keep having conversations about it, especially here at Hatfield. And yeah, we'll figure it out one way or another. Okay, so we're going to start off with a project that I led about 10 years ago now that focused on biogeographic effects of California halibut life history. And you can see some of my main collaborators here. And this is the example where the motive was to inform a stock assessment. And it was through collaborations with folks at Moss Landing Marine Labs, and then a lot of folks at California Department of Fish and Wildlife. So this all started, I was trying to figure out what I was gonna do for grad school and what my master's thesis might be. So I was looking at where there were informational voids, because from the beginning, I was really interested in making sure that my work was useful to somebody. And so I came across the first stock assessment for California halibut, which was done in 2011. And for this assessment, they separated California halibut into 2 stocks, one that was central and one that was Southern California. And there's a number of simplifying assumptions that they had to make. Again, this was the first assessment for the species. And there was some limited information. So one was that we didn't have information about growth and maturity north of Point Conception. And so the stock assessment authors had to make the assumption that things didn't change from Southern to Central California, because we have data from Southern California. They also relied on macroscopic assignments of maturity. And so we know that histological analysis is a lot of times necessary to understand whether or not fish have reached biological maturity. If you saw Claire Roseman's thesis defense, you know a little bit more about that. If we want to move into functional maturity, who is actually contributing to the population, we definitely need more histological analyses to get on the microscopic level. And then natural mortality is something that's really important for stock assessments, and for California halibut, they assumed that it was fixed through time and space for each of the sexes, but they didn't take into account age-specific mortality or mortality that varied from Central to Southern California. So we know Central and Southern California are very different from one another. There's huge differences in temperature. So here you can see the warmer colors are warmer temperatures, cooler. Pretty big difference, north and south of Point Conception. And then also when you look at primary productivity, you can see that Central California is a lot more productive than the waters off of Southern California. And we know that these sorts of things impact growth and maturity rates of fish. And here's an example from California sheephead of growth curves and maturity curves that are just on a very fine spatial scale. So this is the Channel Islands. This isn't even regional throughout California, and you see that there's quite a bit of difference. And one thing I wanted to note is from this study, they ran some models and found that if you really treated California sheephead spatially explicit in a spatially explicit manner, you could actually increase your yield by 26%. And so not only is it about conservation, but taking this sort of information into account can actually provide more opportunities for fishing communities. Okay, so the point of this project was to try to get at whether or not life history traits varied for California halibut north and south of Point Conception. And so what we did is we collected fish all along the Central California coast, and then compared those data with those that were available from California Department of Fish and Wildlife off Southern California. And the real collaborative part was in that Central California region, if we're talking about fishers. And this is a really bad sped up video of me filleting a halibut. But I wanted to highlight a couple things that I found particularly helpful in the collaborations I developed for this work. And so Ed Burrell was just like an amazing human being who ran Capitola Boat and Bait. So this is a rental boat company near Santa Cruz, and he taught me how to fillet fish. I am a vegetarian, so it's not something I had done before. So you should have seen the first few pre-video, but this was huge because then I was able to, one, prepare fish in a way that we didn't hack up the ovaries and have eggs going everywhere, because I was interested in counting those eggs. And then two, I was able to kind of sell my filleting skills to folks to convince them to donate carcasses for my study. So that wasn't all that I learned from Ed, but it was something that was very practical and helpful for me. So do we want to see the end of the fillet video? Or this was also one of the fishermen that I worked with on another project who was taking this video, and he is not much of a videographer. Two other folks that I worked pretty closely with are Curtis and Richard here. They really tried to help me find small California halibut. We failed. We had a lot of good times together, but we could not find little California halibut. And you need all sizes of fish to get these growth and maturity curves. And so that's where one of the collaborations with California Department of Fish and Wildlife came in, where we were able to take samples from San Francisco Bay, which is a nursery habitat for California halibut, and bring them into the analyses. I was just convinced that California halibut were born at 22 inches, which is the minimum size limit. But we're able to work around that. But I very much appreciated all that they taught me about fishing halibut, even though we couldn't find the small ones. And then Travis Tanaka was with, or is with, California Department of Fish and Wildlife. And he really did a lot to teach me how to prepare and age otoliths that's necessary for growth rates. We also tried to find small halibut via beach seine and trawling and all kinds of things. And we did not. Okay. So this is what you get in terms of results from this project. So if you don't trust me, I can show you something better later on. But we did find regional differences in growth and reproduction, but not mortality. And this was important because then we were able to supply region-specific estimates for the stock assessment. And so those growth and maturity rates have been included in the assessment. They've also used our estimates of fecundity that previously was only done in Southern California from captive fish. And so this was the first estimate from wild-caught fish. And I listed total mortality, but it didn't matter, so I don't think they actually used it. And then I just wanted to know Madison Vargas, who's here in the audience, as one of my students working on her master's thesis that is similar, but going to be much more challenging than my master's, where she's looking at biogeographic effects on black rockfish life history, but instead of Central California, she's going from California to Alaska to try to do that. And this is to try to, again, provide information for these state-based stock assessments. A lot of them are assuming values from other areas because they don't have data, California in particular. And this is something that's really of interest to the Pacific Fishery Management Council, because all of a sudden we're having to define stocks. And it's a legal mandate that we're a little bit behind on, and it requires a bunch of pieces of information to try to get at stock structure. Folks lean into genetics and larval dispersal and things like that, but my directive is to try to tell them that life history traits also matter. So we're gonna work on that together. Okay, so now I'm gonna shift gears into the second example that I've got. So we're moving from biology more to ecology. And while I was doing my PhD in Alaska, I really focused on food web dynamics of groundfish. So this was predation and competition in an effort to support ecosystem-based management. The predation component I won't talk about today. That was to directly inform an assessment for Gulf of Alaska pollock. But I'm going to be focusing on the potential for competition between halibut and arrowtooth. And that is the project that Richard Yamada was really heavily involved in. And so we were interested in competition for really this reason here. We saw in the red line shows decreases in growth rates of Pacific halibut. And then the yellow line is increases in abundance of arrowtooth flounder. And those two things seemed inversely related to one another. So we were curious if arrowtooth flounder, because they were just, I mean, they had increased like 6-fold in a few decades in the Gulf of Alaska. So we were thinking maybe they were taking some of the prime habitat or prey that Pacific halibut relied on, thereby reducing their growth. Trying to get at competition in the fields for animals that you cannot directly observe is a challenge. So you'll see a little hand waving later, but it is what it is. We did try to use some spatially expansive long-term data that are available from the Alaska Fisheries Science Center to get at this question. And really, it's just a relationship of these two species overlap in time and space and in what they were eating. We did not find strong evidence for competition. And there were a number of reasons why we thought maybe that was the case. So resource partitioning is the ecological term that we use to try to get at competition. And we thought that maybe we had inappropriate size comparisons of these two fish. So anytime you're looking at diet of fish, you want to standardize or account for in some way the size of that fish, because the larger they get, the more diverse prey and larger prey that they incorporate into their diets, which makes sense. So here is just a simple representation of what we saw from that paper throughout the Gulf of Alaska. And we have halibut on top and arrowtooth on bottom, inverts in red, fish in blue. And so you can see there wasn't a ton of similarities in their diets. And this is according to fork length bin on the x-axis. Now, if we looked at some of the larger fish that we didn't have a ton of samples for, or a lot of overlap in their size, you can see that halibut are eating more and more fish the larger they get, but we don't see arrowtooth flounder that large. And those full blue bars are probably a handful of samples. So then when we looked at the smaller arrowtooth flounder and the larger Pacific halibut, we saw more similar diets. Again, if you're just looking at these like coarse representations. And so maybe one of the reasons we didn't see resource partitioning is that body size isn't really the best size metric to use. And maybe it's some sort of combination of small arrowtooth and large halibut. And I'll hopefully convince you of that here. So when I was a Californian getting ready to move to Alaska, I was like, what are these species I'm going to be looking at? I've never heard of arrowtooth flounder. And I looked them up and I was just like, first of all, arrowtooth flounder is a really hideously ugly fish. But also they have these massive mouths. And if you were to compare the two, so this is some calipers looking at gape width, you can do gape width or gape height. These two fish, I lined them up, they have the same size mouths, but they are very different in terms of body length. And so you can think of how this might come into play if we're trying to compare apples to apples. Maybe body length isn't the size metric that's most appropriate for this type of work. And so that leads us to the question that I was asking along with Richard Yamada and Ambo Dro. And so this was, how does our metric of size affect our assessments of competition? How does what we choose to focus on impact our ecological inferences? So we set out to measure gape and fork length of these two species to one, just say, there is an actual quantitative relationship. And so here you see how gape width varies by fork length for Pacific halibut. And then this is what it looks like for arrowtooth flounder. And to make your job a little bit easier, I've highlighted one fork length bin for the two species, and the mouth is like twice the size for arrowtooth flounder. So you can imagine that they're going to be able to fit bigger items in their mouths at the same body size. And when we went through, these are our sampling sites near Juneau, and we looked at the overlap in fork lengths of these fish. The area in green here shows where there was really sufficient overlap, but it really wasn't that. There were a handful of fish that actually overlapped in body size. If we then converted fork length to gape width, we saw a lot more overlap in the size distributions, which was useful for us for comparisons of diet. So we went in, I had a bunch of students working with me. Again, Madison was one of them, to try to get at what halibut and arrowtooth were eating in Southeast Alaska at the same sites. And so we see here ATF is arrowtooth on the left, PH is halibut on the right, that their diets are similar in a lot of ways, but the key differences are in the invertebrate prey in those warmer colors, and maybe a little bit in the forage fish. If we actually calculated an estimate of overlap in their diets, 0.73 doesn't mean anything to you, but it'll make sense in a second. It's a good amount of overlap. And this is for, I can't remember the body length of this fish, but this is for a single gape width. Now that if you compare that to the samples that we used for the entire Gulf of Alaska, that overlap was 0.13, so it was a lot lower. One of my goals was to revisit those data and make those conversions and see how it changed things. That has not yet happened, but maybe one day. The other thing I wanted to note from this field study is that when you look at the larger gapes for halibut and arrowtooth, you can see that their overlap in diet decreases. So as the mouth size of arrowtooth flounder gets larger, they seem to be eating more of these light blue colors, that's herring, and a little less gatted, so that's pollock and cod. Halibut tend to eat more pollock and cod, and then a greater diversity of invertebrate taxa. And so this comparison of the smaller gapes and the larger gapes makes us think that maybe competition is increased at this latter end of their mouth size. So when we see this resource partitioning or trophic niche separation or whatever ecological term you'd like to use, that's when we think they're trying to accommodate for those competitive effects. Now, here is a bit of the hand wavy part, where we talk a little bit about competitive superiority between these two. And I think that there's a couple things going on here. So exploitative competition is one thing where these cats kind of are displaying it, where the one cat kind of gets to the food before the other one can get to it and just kind of keeps them away. So the prey aren't available to tabby. And this, I think, is happening with arrowtooth flounder because they become of a size where they can eat fish sooner than halibut. So maybe these huge numbers of arrowtooth flounder in the Gulf of Alaska are going in and consuming prey that are important for halibut earlier on, before halibut can actually get to them. And this is one of the things that Richard thought based on his observations at each of these different reefs, where he would see a lot of small a lot of small arrowtooth flounder, but not a lot of halibut. But then when the halibut got big enough, they were able to kind of territorially or some other way keep the arrowtooth at bay. And so this is interference competition where they can kind of, this is where I would think it would be the more superior competitor, but we can chat more about this. Okay, that last piece was, a lot of that really was prompted from observations that Richard had made for years on end in this one location. And so I think the one thing that was really important here was that he was really engaged throughout the process. I think that's the only way that this project was viable. And he was not only engaged, he was like engaged. He developed an app for an iPad to collect data. So one of the concerns always when working with fishermen is, how do I get the data that we need without taxing them? Because they are plenty busy, especially when you're dealing with sport fishing lodges where they have guests and they're filleting fish and they're doing all kinds of things. And so the iPad app was one way to simplify things for them. The other thing is that Richard just got everyone involved. This is his granddaughter taking data. We didn't use those data. But he just like had a way of getting even his most like curmudgeon-y captains interested in the work. And just as a side note, he built that table right there just for this project at his lodge. So it's just the investment that he had in this was just great. And it was because he had seen this decrease in growth of halibut really impact his business? Okay, so the last example I want to show you today is how we can go about improving species distribution models to try to get at effects of climate on groundfish. And this is again in Alaska. And so I don't need to tell anybody here that we need to try to get a handle on effects of climate change. This is particularly noticeable in Alaskan waters where we've seen the greatest warming. Species distribution models are one way that we can try to get a handle on climate effects. These types of models really try to quantify distributions and densities of fish as a function of the environment. And so this large map would be predictions of the probability of occurrence of some species in relation to a variety of environmental covariates. So this might be temperature, depth. A few of these are presence, absence of structure-forming invertebrates. But the problem is that the SDMs that are used for management, and particularly essential fish habitat in Alaska, are static in time. So what they do is they rely on these long-term mean conditions to try to understand where fish are. And if we want to take a more climate-informed approach, we have to not only account for spatial variation, but we have to bring time into as model covariates as well. And so that's where our focus was. Oh, and forward going, I'll probably interchange these names, but these conventional SDMs we're referring to as static, and these more climate-informed SDMs are what we're calling dynamic. And so the question was, do these climate-informed models actually improve our ability to hindcast species habitat associations, so getting distributions and densities fully in the past? And then can climate-informed models increase our forecast skill for near-term responses to climate change? And for this project, we really thought they have to. There's no way they could not. And there's some interesting areas, I think, where we can continue to develop these models because that surprisingly wasn't the case. But what we did for this is we took bottom trawl survey data from the Alaska Fisheries Science Center, ran some GAMs, generalized additive models. I'm not going to talk about any of the methods, but if you're interested in retrospective skill testing or some of the performance metrics, let me know. But we essentially ran 600 and something models to try to get at hindcast performance and forecast skill. And so we focused on pollock and arrowtooth flounder in the Bering Sea. And basically there's a ton of data for these two species. We had grandiose plans of doing like 6 or 7, and we got left with 2. But here is the static representation of what we might say is important habitat for pollock in the Bering Sea. And EFH, the part that we really use in spatial management in Alaska, is that dark blue color. But we are increasingly interested in these finer levels of habitat importance, going all the way up to these yellow areas that we're calling hotspots. And so this map relies on long-term mean environmental conditions from the 80s to 2010s. Well, 2020s. So then we wanted to see how this map compares to a dynamic approach. And so here's a subset of predictions from the dynamic models used to estimate habitat importance. And you don't really want to pay attention to what's going on in the northern Bering Sea with these big yellow blobs because we don't yet trust those data, but everything else I feel like is reliable. And you can see that even though the static models generally reflect the more important habitat from one year to the next, those yellow hotspots move around quite a bit. And that was the same for arrowtooth flounder, despite the fact that this is a very different species, plays a different role ecologically in the Bering Sea, isn't, hugely targeted like pollock is. It's still, the interannual variability was generally captured by the static models, but the dynamic climate-informed models, when looking fully in the past, were clearly the way to go. So these climate-informed models did great in the past. I'm not going to show you the breakdown of forecast skill, but going into the future, they started to break down. And it was really surprising that the static models had greater forecast skill going 5, 10, 15 years into the future. Similar to the forecast skill for the static models was what we call a persistence forecast from the dynamic models. So just assuming that the last year of the data you used to fit the model, that relationship kind of carried out. you could use those, but it was really very interesting that if you're interested in forecasting, you can't just rely on whichever model did the best in the past. And so we developed a few recommendations for SDM users. Plenty of people use these types of models. And so we're really kind of advocating for something like retrospective skill testing, which is what we use to try to figure out what model was going to do best going into the future. And I guess I have to explain that part a little bit. So how we were able to estimate forecast skill is we were predicting into the future, but it was fully in the past where we had data to compare it to. So hopefully that makes sense just verbally. But you have to go through that process to understand how things might change from hindcasting to forecasting. We also think some issues with temperature came into play with the models not really knowing what to do with hugely anomalous temperatures in the Bering Sea. And then we're interested in seeing how these models can be improved if we bring ecological covariates into the mix. I'm going to skip this and go right here. So Perry Gerson is my other master's student. She will be actually trying to figure out how we can get a better handle on some of these ecological covariates. And she'll be using the diet data from the bottom trawl survey to kind of treat predators as samplers and run these species distribution models to try to get at prey availability throughout the Gulf of Alaska. And then the goal here is to try to use those prey predictions as a proxy for habitat quality for key groundfish, and then incorporate them into the SDMs for these species that we actually identify essential fish habitat for. Now I'll go back to this. So Jody Pirtle is somebody that I've worked closely with for this work. She is at the Alaska Regional Office, and this is the group that really handles EFH and spatial management. And so they're taking some of these techniques and improving the models that they use for management for it. And we're hopeful to continue to iteratively do that for the Bering Sea and the Gulf of Alaska. That's the EBFM piece. Okay. No, it's the climate ready fisheries piece. It's both. So I went through some examples of how these projects really, because of the collaborative component, helped us inform fisheries management in a variety of ways. And one thing that I would love to do is figure out how I can work more closely with you folks here in Oregon. A lot of my connections are still in Alaska. Before that, they were California. And I am really trying to get to know you folks here to see what projects are locally relevant. And this is the group of people at the Marine Resources Program that I am connected to in one way or another right now. Those of you who are in this photo, I would love to figure out really how we can better work together, but also beyond MRP. I'd love to chat, especially folks who spend time out on the water to try to develop some research questions for Oregon. And I will just throw this up since I have a little bit of time. There's another project that I didn't talk about. It involves simulation modeling with food webs in the Bering Sea and the Gulf of Alaska. This is part of two big programs, ACLIMB and GOA-CLIMB. So these are groups of people who use projections of environmental data to try to understand what may be happening with different climate and fishing scenarios in the future. We're going to be using a MISR model, so it's a size-based food web model to try to get at some of that and compare that to other ecosystem models. And so we've got funding for a PhD student to start next fall on that. So if you know of anybody who might be interested, preferably they have a little bit of quantitative ecology background, but that person will be co-advised by John Ream, who's in the food habits group at the Alaska Center, and myself. Okay, I'll leave it there.
All right, any questions from the audience or online?
Thanks, Cheryl. That was awesome to hear about all your cool work. I'm really curious about that last piece on the SDM predictions using sort of climate informed variables. And it's pretty intriguing that the more dynamic climate informed ones sort of crapped out in the predictions, it sounds like. I'm wondering if you could expand a little on why. Like, is it because of sort of anomalous conditions that are not in the training data, but if you're testing them by predicting to the past, then I would want, anyway, I'm curious if you can elaborate on why you think the predictions fall apart when including more dynamic data.
I'm trying to get there with all my extra slides here, but I don't see it. Yeah, so there are two big things with so we only looked at 2 species, so it's really difficult to get at trends. Oh, okay. It is going on its own. Okay, so this is the forecast skill that I didn't show you. So the models generally, like regardless of what model we used, did pretty well for arrowtooth that's on the right. Not that great for pollock, even at the start, at the 5-year time frame. And the pollock piece, I mean, it is one of, if not the largest single species fishery in the world, and we didn't incorporate fishing effects. So I can imagine that might increase our forecast skill for pollock. There's also those ecological variables that we're just missing. But that doesn't really explain the difference between the static and dynamic models for arrowtooth flounder. And so one of the things we think for arrowtooth flounder, because you see this, the static models are doing pretty well out to 15 years and the dynamic models kind of decline, we think that maybe there's a relationship with temperature that actually is representing something else, like prey availability, and that breaks down with time. But also it was this, there we go. It did a lot of exploration of temperature because I just did not know what was happening here. And this was the biggest dynamic variable that we had, in addition to just like latent variation of time and space. And so you can see here with the data that we fit the models with, pollock and arrowtooth on the left, those are the temperatures that we observed where those fish were. And then the ones that we used to forecast are on the right for static and dynamic models. And so for the static models, you can see there's a lot of temperatures outside of the range that we saw for arrowtooth flounder. And then those are exaggerated when you take year-specific estimates into account. And so I can't tell you if that's like a product of using GAMs, because we only used a single modeling framework. And so maybe other models can do a better job, totally possible. But I think that's part of it, that the models also just don't know what to do with that new information. And we did a lot to try to like safeguard against that with how we parameterize, or sorry, how we configured the models, but that's where we're at.
Well, super interesting, thanks.
Yeah.
No questions online?
Sorry, I was about to drop it. Sorry, I actually just following up on Dawn's, I guess maybe can you dive in a little bit into fitting the dynamic models? Because like when I learned like baby SDMs, there was always the warning of like needing to take longer climatology so you don't bias your predictions based off of like decadal trends. So I guess I'm just curious if you can maybe like, I'm sure you thought of that. So I guess I'm just curious what goes into the dynamic GAM like building process.
Yeah, I pushed the arrow. So we'll see if it shows up, because there's a slide that shows all the different variables and how they're grouped. But so these are pretty simple dynamic models in that we really relied on temperature. And so temperature doesn't get at everything. We, at the time, some of the ROMS models that we were using, I think like stuff like pH and whatever wasn't as developed. And so we just opted to stick to temperature, but they have been doing a lot of work on that front so that you can bring those sorts of things in. Zooplankton abundance, I think, would probably be useful for another iteration of this. I tried that too. Oh, that went the other way. There we go. Well, that's the process, but okay, now this is where it's going to take a second to load. Yeah, and so then the other thing besides temperature that was really unique for the dynamic models is that we accounted for temporal variation for the simpler ones, and then that and spatiotemporal variation for the most complex. And that's the one that we thought was going to do the best because we're accounting for a lot of the variation. So you can see kind of the breakdown of increasing complexity in those little outlines. Oh, cold pool extent too is a huge thing in the Bering Sea, as you know. So we incorporated cold pool extent as a spatially varying coefficient. So tries to get at the annual index of cold pool extent and how that might change spatial distributions as a result, because it essentially serves as like a big barrier in the middle of the Bering Sea that a lot of fish won't cross. Did that answer it?
Yes.
Do you have
Thank you.
follow up?
No.
Okay.
Any other questions?
No. Gotta be that way.
No, this is well, actually, maybe it's not an easy question, but it has nothing to do with fish. No, but it goes back to the very beginning of your talk where you talk about essentially co-production of knowledge with fishers, stakeholders. And of course, this is something we work at all the time up in the Arctic. But thinking about like you focusing on a couple of your fisher guys, because they're often guys, and like I've got a number of people that I always reach out to.
Mm-hmm.
I think it'd be useful to think about how do you find those people? How representative are they of the industry in general? Or is it just getting really lucky? Like, what is your advice to students who want to try and co-produce knowledge, whether it's with whalers or fishers or any resource user?
I will speak to my own experience, and you can take that as advice if you'd like it. But casting a wide net, I think, is really important. And then spending time and working on building relationships. So like I spent a ton of time hanging out at fillet stations and the like boat rental shop and getting to know people on a personal level. And one great example actually is Curtis, one of the guys who I showed who took me out trying to find little halibut. He was so uninterested in talking to me at the start. He was just like, here's another person out here trying to whatever. I mean, there's a lot of assumptions that I want to like close the fishery and things like that. Like that's my end goal. And he was a small scale commercial fisherman and he avoided me forever. And then he became my personal goal to like befriend. And I think it was just the fact that he saw my face so much and that he saw that I was developing friendships with his friends. And then we would just start off with casual conversation. And then like by the end of it, he was just, I mean, he was taking me out on his boat, like just to fish for my project. So I think it's just time and energy and realizing that we're all people. And the more you develop a relationship, the better it will be for the science, I guess. Vaughn's got an addition to that.
I do. We have a very well documented history of research collaborators being disconnected from the results of that research.
Right.
Can you speak to the importance of that and potentially the reactions from your collaborators when they found out the findings of your studies?
Yeah, so that I think is a big difference between outreach and engagement, is bringing the information back to the people that you have hopefully not just extracted it from, but worked with to collect. And reactions of the work. So like back when I was doing my master's, I would like send them Christmas cards with like updates of how many fish we got and like that sort of thing. So that wasn't really update based because we all know that all comes like year 3 of the master's. I don't think there was a lot of surprise with what we found. I mean, we didn't really have anything for Central California. So there wasn't a lot there. And then there was a bit of delay for when it was actually used because I wasn't that end user for the assessment. So that's not an answer. For the competition piece, one thing that we've always struggled with is like the time it takes to have results, because scientists seem to take forever to do anything and fishermen are like, what the hell is the product of all this work that we've done? So that's one of the struggles, I think. And then when you're dealing with like complex ecological relationships, like it isn't clear cut and people want a yes or no answer. And so that's a bit of a struggle. Trying to think of any other examples. I also was part of a collaborative fishing program that I didn't talk about that did MPA monitoring, and there was a lot of engagement there every year. We'd talk about the results. What do you think is happening? Do you think it's actually the MPA or something else? For that type of project, a lot of the people had to be open to MPAs to some extent, but then there were other folks who were like, if you're gonna use these data to decide if these things are working, I want to be a part of it. I want to have a say. So that did not answer your question, but like that's been my experience.
I appreciate you sharing your experience. Jumping from that though, can you speak to like the potential of continued work with those collaborators had you not re-engaged them, or if it had been like a simple extractive process? Is getting to your point over there is like
Burning the bridge.
correct.
Yeah,
Okay.
yeah, for sure. Yeah, I mean, parachute science is a big problem where you go in, you extract, and you say thank you, and now they don't hear from you again. So the next time you try to go in and run a project, like you're gonna be met with more opposition than you would have if you fully engaged people throughout and they saw the fruits of their labor, really. And I think that just gets back to realizing that we're all people and we kind of all want to see the fruits of our labor. But it's not just, again, about communicating it in one direction, because there's so much that you can gain from, again, those people who are out there all the time. And I have learned much more from fishermen than I have in textbooks, like for sure. And so it's that mutual exchange of information, I think, that strengthens your work in the end too. So wins all around if you bring it back.
Thanks. You mentioned potentially compensating fishermen for their time and efforts in participating and writing that into grant proposals, et cetera. I'm curious, in your experience, I mean, do you have experience with doing that? And if so, do you feel like that has made a difference in the number of people who've been participating and collaborating with you, or the amount of time they have been willing to devote to your project and helping out?
I haven't seen like a cause and effect like you're describing. The example that I have where we wrote somebody into a grant was Richard Yamada, and like he would have done it either way. So, and it was just like contract services was how I think we wrote him in. I think part of that maybe covered costs with the app, but then he also went out of pocket for that too. So again, like Richard's not the best example because he's just like, I think he would have been a scientist. Like he's had like 5 careers and like scientist would have been one of them. And like with my master's, I had no money for myself. So I had no money for anybody else. So I would like buy burritos and beer and stuff for people and then like be very verbally appreciative. So yeah, I haven't seen a direct cause and effect. I don't know if that's because there isn't one and people are gonna choose to put their time and energy in regardless. But like if they're not doing this as part of their business, like also Richard was doing, you only have so much time. So I'm like giving a lot of non-answers today. Nuance, it's not black and white. Jessica.
Cheryl. So my question is, you talked about wanting to do projects that have that nexus between science and management and are applicable to management decisions. And so I was wondering if you could speak to how you pick a project, knowing the timescales in which science occurs and knowing the timescales in which management actions occur. How do you be strategic about which management actions and projects to take on?
Maybe you choose to do Black Rockfish life history right after the assessments have concluded. So then you buy yourself a few years. And I guess that's what I did with California halibut too, was like, who knows when they're gonna do a next assessment, but we'll have the information when they do. But I think that's something that like I really want to work with you folks on at ODFW because it is such a challenge. And I think maybe then it's more like my time or postdoc time for things that like need to happen right now, and then things that could have a little bit of a longer time scale are more like better suited for student-led research. Anybody online? No? Okay.
Please join me in thanking our speaker today. And if you haven't signed in, please sign out. And the recording of this will be available on our website. So thank you again for joining us and see you in 2024.